Question number 32. So they say during the gametogenesis is the formation of gametes. So in this person, during the formation of gametes, a gamete has been formed with 24 chromosomes. And then they say this gamete was fertilized with a normal gamete and a child was born. Which of the following best explains this process and its result? So this gamete having 24 chromosomes will fuse with a normal gamete. A normal gamete will only have 23 chromosomes. So when they fuse and form a zygote, the zygote will have 47 chromosomes. 24 plus 23, 47. But a normal human cell should only have 46 chromosomes. So obviously there is one extra chromosome because of that gamete which had 24 chromosomes. So always if a cell has one more chromosome or one less chromosome, that condition is called aneuploidy. So because of that, answer number 2 and 5 are wrong. So if a cell has one more chromosome or one less chromosome, it is aneuploidy. And then just see, because this final cell formed has one extra chromosome, that means it has 3 copies of the same chromosome. Just consider the gamete having 24 chromosomes has 24 because it has an extra copy of some chromosome. So if the gamete having 24 chromosomes has 2 copies of the chromosome, the other gamete has 1 copy of the chromosome. So that is how these gametes are formed. So we know the father's gamete has one chromosome from each type. The mother's gamete also has one chromosome from each type, and then they fuse, and the zygote has 2 chromosomes from each type. That is the normal situation. But in this situation, because in the gamete with 24 chromosomes, there is an extra copy of a specific chromosome, the final cell formed has 3 copies of a particular chromosome. So when a cell has 3 copies of a particular chromosome, the normal condition should be 2, but here there are 3 copies of a chromosome. So because of that condition is called trisomy. So because of that, answer number 3 and answer number 4 is wrong. So the answer is answer number 1. So we know Down syndrome is aneuploidy and trisomy, and in the case of Down syndrome there is an extra copy of the 21st chromosome. So the answer is the first one. Then consider the 33rd question. So here they say during replication of DNA, a cytosine molecule had been added instead of a thymine molecule in a gene. Then they say this muted gene produced a peptide with the same amino acid sequence as the gene before mutation. So the important point is, if the amino acid sequence, if the final amino acid sequence doesn't change, that means it is a silent mutation. So the answer should be answer number 2 or answer number 3. And here they have used the word instead. That means, according to the question, a thymine molecule has been replaced with a cytosine molecule. So if a particular molecule has been replaced, that is a substitution. So according to the description, a substitution has occurred and it is a silent mutation. So answer is the second one. Then 34th question, DNA polymerase obtained from thermophilic bacteria is used for PCR because. So we know in this PCR we are using something called Taq DNA polymerase. So this Taq DNA polymerase is obtained from a bacteria called Thermus aquaticus, and this bacteria is thermophilic. That means it is not sensitive to high heat. It can survive during high heat. So during PCR we have to heat it to a very high temperature to separate the two DNA strands. So that is why we use the polymerase we obtain from this thermophilic bacterium, because it will still be stable at high temperatures. It won't be damaged at high temperatures. So the answer is answer number 3. DNA polymerase is stable at high temperatures required for the separation of DNA. Then question number 35, it's all. Question number 36, dry Patana grasslands in Sri Lanka are found in intermediate zone and wet zone. That is a theory point. Answer is answer number 1. Question number 37, which of the following is not an environmental service value of biodiversity? So this is in page number 90. So in under biodiversity, we discussed about something called the environmental service value, and under that you can find regulating climate, recharging groundwater, water purification, preventing of soil erosion, and other examples are given. So this helping disaster management is not an environmental service. So that is the answer number 4. Question number 38, which of the following does not contribute to global warming? So factors that contribute to global warming are basically the increase in carbon dioxide levels in the atmosphere, then deforestation, and the destruction of phytoplankton. So when the ozone layer is depleted, phytoplankton is destroyed, and that will contribute to global warming because the amount of carbon dioxide in the atmosphere will increase. And they have given other points as well. So if the level of methane increases, that will contribute to global warming. Apart from that, industrial gases and other types of gases are mentioned. In addition to that, this water vapor also contributes a little to global warming. So water vapor also acts as a greenhouse gas, that is mentioned in the resource book. So the answer is answer number 4. Growth of phytoplankton will help to decrease global warming, not contribute. So answer is the 4th one. Question number 39. Which of the following statements is correct regarding the culture media used to grow microbes in the laboratory? The first statement is wrong. So they say agar is used to provide a suitable pH range. That is wrong. Agar is used as a solidifying agent. Second statement, glucose is used to prepare culture media to grow fungi. That is correct. That is the answer. So in our syllabus, we learn about 2 types of culture media, nutrient agar and potato dextrose agar. So nutrient agar is used to grow bacteria. Potato dextrose agar is used to grow fungi. In this nutrient agar, you cannot find glucose. In potato dextrose agar, you can find glucose. So because of that, we can say glucose is generally used to prepare this culture media to grow fungi. So second one is the answer. If we consider the other statements, third statement says culture media for bacteria prepared using potatoes. That is wrong. For bacteria, it's nutrient agar. Potato dextrose agar is for fungi. Fourth statement, any microorganism can be cultured in a culture medium. That is wrong. Some microorganisms cannot be cultured, and they are called non-culturable microorganisms. You can only culture some types of microorganisms. Fifth statement, sodium chloride is usually added to all culture media. That is wrong. So if we consider the media we know, you can find sodium chloride in nutrient agar, but you can't find sodium chloride in potato dextrose agar. Question number 40, coliform bacteria were detected in a water sample obtained from a river. Drinking untreated water from this river may not likely to cause. Okay, so the theory behind this is if coliform bacteria are found in our gut. That means basically the stomach and the intestines in our gut, you can find coliform bacteria. They are relatively harmless in our gut. So because they are found in our gut, you can find them in feces samples as well. So that means if you test a water sample and if it contains this coliform bacteria, that means that water has been contaminated by feces. So this is used as a test also to see if a water source is contaminated by feces. They will test for coliform bacteria. If coliform bacteria is present, that means there is fecal contamination. So if we consider these answers, we can see typhoid, cholera, dysentery, paratyphoid, and in addition to this there is something called gastroenteritis. All these are waterborne diseases. That means they can spread through water. So you can say that if you drink water from a contaminated water source, you can get all those diseases, but tetanus is not a waterborne disease, so you cannot get that. So the answer is answer number 5.